here we have Jack and Jill. They're in a loving marriage relationship and they want to have a baby. But there's a problem. There's a 1 in 4 chance that their baby will be born with cystic fibrosis. In order to eliminate the risk of having a child affected by cystic fibrosis, they decide to undergo a very expensive procedure, preimplantation genetic diagnosis. At the beginning of preimplantation genetic diagnosis, they need to undergo in vitro fertilization. So several eggs are removed from the mother, and sperm is collected from the father. The sperm are implanted into the eggs, and fertilization occurs. Once the eggs have been fertilized, and we have several embryos, it is time to perform the biopsy. The biopsy can be performed in any of the 3 stages: polar body, cleavage, or blastocyst. But in order to perform the biopsy and see whether or not these embryos carry the risk factors for cystic fibrosis, the doctor needs to take a closer look at the DNA. But rather than make the DNA bigger, the doctor can multiply the number of important strands via polymerase chain reaction, or PCR. The first step of PCR is a target sequence is chosen on the DNA. Then 2 DNA primers that were synthesized for each strand are added to a mix. Third, modern techniques use tag polymerase to add nucleotides to the new strand. The 4th step is all 4 DNA nucleotide building blocks are added in sufficient quantity. The sample is then heated to a temperature of up to 98 degrees Celsius to separate the complementary strands. This step is called denaturation. 6th, the sample is then cooled. During the cooling stage, the synthetic primers found complementary sides on the separated DNA strands. This process is called primer annealing. 7th, the polymerase extended the 2 primers in opposite directions. As a result, 2 daughter DNA appeared, and then the process is repeated until more DNA strands are created. By the 30th cycle, there can be up to 1 billion copies of the DNA. Now that we have a lot of the same DNA strands, we can separate its components via gel electrophoresis. Now, when looking at the gel electrophoresis output, if the doctor determines that it's free of risk factors for cystic fibrosis, it is a winner, and the embryo is implanted into the mother. However, there is another way we can look at the DNA. We can go fishing. Fish or fluorescence in situ hybridization is a way to tag certain DNA sequences in order to detect unwanted defects. Sequences of DNA are exposed to fluorescent probes, or dyes, which are synthesized from the genes we wish to detect. The probes will bind with and illuminate the DNA strands that they are similar to. Once we find an embryo with DNA that we desire, just like in PCR, in this case it'll be the embryo that is not tagged by the fish, the embryo will be implanted in the mother and will eventually become a child.
That was